Okay, so now we're going to turn to the techniques that track electricity instead of blood. So these are the fast techniques. And we'll talk about electroencephalography, or EEG, and magnetoencephalography, MEG. And again, I want to give you a little bit of an idea how long these techniques have been around. So EEG was invented in 1924, so it's been around since the 20s, and MEG has been around since the early 70s. That gives you a bit of a historical timeline. Okay, so these techniques are really important for the neuroscience of language because they have the temporal resolution that we need in order to characterize the rapid temporal dynamics of language processing. So both of these techniques have a temporal resolution at the millisecond level, and that's the correct temporal resolution for language processing. And so, as we've already learned, neurons communicate with each other by sending thousands of times per second by sending each other tiny electrical impulses. And when populations of neurons are connected into networks and fire in synchrony, then it's the dynamics of these large synchronous activities that we can detect non-invasively outside, either on the scalp in the case of EEG, or actually outside the head in the case of MEG. Okay, so the main source of the signal for both techniques is postsynaptic current flow along the dendrites of nerve cells, and in particular, certain shaped nerve cells more than others, so pyramidal nerve cells, but you don't have to worry about that too much. So here we have our neuron that we've already taken a look at. So along the axon, we have the action potential rushing through, and then it's going to make contact with the next neuron, so not completely, but there's this little gap between the axon and the dendrite of the next neurons we call the synapse, and then the postsynaptic current flow along the dendrite is what these electrophysiological techniques mostly capture. And so this is just zooming in a little bit more. So we have our axons meeting the dendrite, and then the current flow along the dendrite is what we would be measuring. Okay, so that's the electricity rushing through your neurons. So now what we need to know is that an electric current generates both an electric potential and a magnetic field. And so in EEG, it's the electric potentials that we measure. So EEG obviously is probably already familiar for everybody. It's almost in any doctor's office. We can use it to measure other organs than the brain as well, so like the heart. And then MEG is what measures the magnetic fields. So here's a picture. So we have an electric current. So that's a cartoon representation of your neuronal current. It will create a potential out here and here. So you'll have a negative potential here and a positive potential here. And then around the current, we have a magnetic field rotating. So now if we're looking at the picture on the scalp, we would have the current here creating that electric potential. So negative potential here, positive here. And then around the current, we also have a magnetic field. So those are two different manifestations or two different correlates of the underlying electrical activity. So we can measure both of these reflections of the current activity. And so now, in order to be able to think about the relationship between the current and the magnetic field, we need to keep the right-hand rule in mind. So what's the right-hand rule? It tells you that if you take your right thumb and then wrap the fingers around it, the direction of the magnetic field, assuming that the thumb is the current, so the thumb is now pointing upwards, so the current would be flowing in this direction, just like in the slide here. And now the magnetic field would be rotating in this direction. So it's the fingers here that are showing that it's going this way. Okay, so rotating this way. So that's your right hand rule. So that's the relationship between the current and the magnetic field. The EEG and MEG setups look quite different, and by the end of this video, you'll fully understand why. So EEG is measured from the scalp. So here's one particular type of EEG cap that has the EEG electrodes right on the scalp. Whereas in MEG, the sensors that make the measurement, they don't actually touch the subject or patient at all. So they're actually lying in this dewar, and the dewar is filled with liquid helium. That's something that's going to help us to measure these really, really tiny magnetic fields. Okay, so what does the data look like? Well, again, for EEG, you probably have some idea already. So here we're imagining that we have those EEG electrodes on the scalp, and each one of them is going to be measuring the voltage of the electric potential on the scalp. So here's a very high density EEG array, lots of electrodes on the scalp, and you get the squiggly line for each electrode representing activity at that particular site. So that's what raw EEG data looks like. And raw MEG data looks very similar. So again, here each line shows the strength of the magnetic field in this case over time. So the x-axis is time here. And again, here you can see, so now we're not measuring from the scalp, but the MEG sensors are actually a little bit further away, not touching the patient or subject at all in the way that in EEG they would be. Okay, so from this, in this continuous type of raw data, the measurement reflects all the electrical or magnetic activity that is happening at that sensor site. And a major challenge for these techniques is to separate signal from noise. So signal is what we talk about when we're talking about the brain's responses to the stimuli that we actually want to study. So if I want to study, well, how does the brain find the meanings of words, then I'll be presenting the subject with words, and I'm really interested in the activity that's reflecting the word processing, but I need to somehow extract that from all the other stuff that's happening. So what's all the other stuff that's happening? What's the noise? Well, of course, there is something that you could call brain noise. So if you're an experimental subject, you're just lying there, and especially if the experiment is boring, which unfortunately lots of experiments are, you might be thinking about all sorts of other things as well. And so then the signal that we're collecting is going to reflect all of that. You're thinking about like, oh my gosh, I haven't submitted my taxes. Oh, here's another dog that I have to read, etc., etc. So it's a combination of all those different things. So that's one source of noise. But then your body movements also generate shifts in electric fields. And of course, the closer the body part is to the brain, the more those shifts are reflected in our brain recordings. And one body part that is moving, even if you're trying to be really still, is your eyes. So we can't, if our eyes are open, we can't go without blinking for too long. We can keep our mouth shut much more easily, but the blinking is probably going to be happening. So we need to worry about those kinds of what we call artifacts. And then finally, and this is more MEG specific, in the environment, there's lots happening. These labs are usually either in hospitals or in university buildings. So there are lots of shifts in electric fields in the environment. So like if your MEG lab is next to an elevator that's going up and down, well, that's a lot of electromagnetic noise. And even smaller things give you lots of noise. So the fundamental question then is like, how can we capture the signal from the noise? So the raw data is going to reflect all of this. Okay, so let's start. How do we kind of get rid of the brain noise first? So the kind of age-old technique for trying to extract the event-related brain activity from all the other brain activity that may be happening is what we call averaging. It's pretty simple. So the idea is that we just average together all the instances where the brain is processing the stimulus that we're interested in, and that kind of enhances the activities that are just related to that stimulus, and everything kind of gets canceled out. So here's a kind of old-fashioned picture of an EEG setup. So same basic logic applies to MEG analysis. And so here's your, your experimental subject is reading the word dog. And so you're running a very basic experiment where you're just interested in the brain's responses to the word dog. And so now these are the presentations of dog. You're going to be reading dog many times. And what you're going to mark in your EEG data is the onsets of these events. So every time that dog is presented, you're going to send a little pulse so that you can also, so that you can then find each instance of dog from the raw data. And then you're going to average all of those, what we call epochs, together. So I'm going to clip out this part of the data and this part of the data and this part of data and average them all together. And now everything that is not time locked to the onset of dog, and we'd expect the, like your visual response to dog always to kind of happen at the same time. So everything that is not time locked is going to be varying randomly and will probably be canceled out. Or at least that's the hope. And so as a result of this, we would then get the average, or the average so-called event-related potential in the case of EEG. And if it was MEG data, we would just call it an event-related field, the field reflecting the fact that it's an MEG field and not an electric potential. So here, even though the continuous data may be kind of messy and hard to interpret, in the average, you should now be able to see responses that are very characteristic of that specific type of stimulus. Okay, so here, activity that's time locked to the presentation of the stimulus is enhanced, and all other activity that varies randomly is supposed to be canceled out. It's the basic idea. Okay. And there's many other ways to also discriminate between brain noise and signal noise, but this is the sort of most classic, most traditional one, and we can run pretty far in this course with just this idea. Okay, what about the artifacts? So what about your eye blinks, maybe your cardiac, your heart is beating also, and it's not too far from the brain? Well, this is not super exciting, so I'll just tell you what we usually do. So we can do something that's called artifact rejection, and that just means that we clip out the parts of the data that are contaminated by these artifacts. You just throw them out, don't include them in your analysis. We can also model the distributions elicited by those other activities if they're pretty regular. So for example, we know very well what an eye blink looks like in MEG data or in EEG data, and we can use, for example, independent component analysis to then remove that from the data. So that's something that we can do as well. Of course, artifacts that are more surprising may be harder to figure out, but there's a bunch that are pretty regular that we know how to deal with. Okay, here's then a harder one. So especially in MEG, and so mostly in MEG, we have to deal with environmental noise in very fancy and sort of expensive ways. So the fundamental nature of the MEG signal is that the magnetic fields that we're trying to capture are super tiny. Here's a little scale that shows you where the human brain is. So it's down here, very, very tiny signal. And almost everything else that happens is a much, elicits or causes a much bigger magnetic field. So These are just, you know, human eye, fetal heartbeat, skeletal muscles, human heart, lung particles, all of these are now the body noise. The biomagnetic fields are larger than what the human brain puts out. And then the urban and environmental noises, they're, you know, many orders of magnitude stronger. And here, so the Earth's magnetic field obviously is also much, much stronger than your brain's magnetic fields, but that's, of course, it's static. Okay, so how can we then capture this very, very small signal? Well, there's three main ways that we do that. So first of all, our sensors have to be really sensitive. So our sensors are superconductive, so they involve, you know, fancy physics. And one component that, you know, any user of MEG kind of encounters and has to deal with is that the sensors lie in liquid helium. That's part of what makes them superconductive. So here we see, you know, so there's your head with a brain in it. And then here's this tank or the dewar that is filled with liquid helium. So very cold, like, you know, that should be minus 269 Celsius or minus 452.2 Fahrenheit. And so that's part of the our ability to capture this tiny signal. In addition, the whole system is inside its own little house. So it's inside a magnetically shielded room. And the magnetically shielded room is designed to block magnetic fields from the environment. So that's already something that's supposed to lessen the environmental noise, you know, at the sensor. So it's kind of in a protected environment. And then finally, if there is environmental noise creeping into the room, we try to capture it, we try to measure it, and then we try to remove anything that's correlating with that signal from the data analysis. So in addition to these MEG sensors here close to the brain, we also place so-called reference channels a little bit further away, so far enough that they're not capturing the brain signal, but they would be capturing anything else that's coming from the environment and sort of creeping into the room. And so we measure the activity at those reference channels, and then in data analysis, anything that is correlating in the actual MEG measurement with the measurement that we took out here is going to be taken out. And that way we feel more confident that the MEG recording is not reflecting, you know, the elevator nearby or something like that. Okay, so lots of procedures and properties of the lab allow us to try to capture the signal from the noise with the measurement reflecting all these different things. Okay, so now so far I've talked about EEG and MEG in a pretty parallel way. And in fact, they are very much complementary techniques, but they do differ in certain ways. And so in particular, they differ in price, they differ in resolution, and they also differ a little bit in their coverage. And by coverage, I mean what brain activities they're able to pick up. So let's talk about each of those in turn. First of all, price. So there's a big difference in price. So EEG is much, much cheaper than MEG. So you can certainly get an EEG system for less than 100K, but MEG systems, because remember you have to buy the room too, the magnetically shielded room, are more like a couple $1 million. So for this reason, EEG is much more common and available than MEG. And it's really so it's sort of tackling all of the challenges of capturing the tiny magnetic fields is what makes MEG more expensive. Okay, so resolution-wise, temporally, they both have excellent and parallel temporal resolution. So for both of them, the temporal resolution is in the millisecond scale. So we can make the measurement millisecond by millisecond, and that's good enough for pretty much any cognitive neuroscience question that you might have. However, when it comes to spatial resolution, MEG has much better resolution than EEG. So with MEG, we can figure out where the underlying current that's generating the magnetic field actually is in the brain. And it's that underlying current that, of course, is your brain activity. So why do we have this difference? We have it because for the purposes of the electric potential, all the different tissues of your brain, there's a lot of different structures, and your skin and your scalp, all of those different structures differ in their electrical conductivity. And what that means is that the potential gets distorted or kind of morphed many different ways before it reaches the electrode outside your head, so on your scalp. And that distorts the electric potentials enough that it's actually pretty difficult to figure out exactly where the underlying current is coming. However, for MEG, the magnetic fields pass through the different structures of the brain and your head undistorted. So there's not this conductivity issue. And for that reason, localization of the underlying currents, so your actual location of the brain activity, is much more straightforward and in fact a routine part of MEG data analysis. And we get spatial resolution in the order of around a couple of centimeters, but it does depend a little bit on the depth of the source. So the magnetic field strength drops off very quickly the further away you get from the current, and so that's something that's a factor. But what we routinely do with MEG data is use source localization algorithms that look at the magnetic field that's recorded by the sensor array, and then we reconstruct exactly where in the brain that magnetic field has been generated. Okay, so coverage-wise, for MEG, current orientation matters in a way that it doesn't for EEG. And so now what I mean is that the orientation of the current in your brain, so of course the currents could be oriented in many different ways. So let's look at this a little bit. So here's two different currents. One of them has a radial orientation and the other one has a tangential orientation relative to the skull. So now in EEG, the radial source would give you a nice juicy positivity right here on top of the head, if we imagine that that's the top of the head, whereas the tangential source would also give you positivity kind of on the top of the head and then a corresponding negativity out here. I didn't put the negativity to the radial source because it would, if this is kind of in the middle of the head, probably wouldn't be captured very well out here. What about the MEG situation? So here, let's start with the tangential source. So here, if we imagine that there's a rotating magnetic field around this current, we're going to be able to capture it really nicely with our MEG sensor that is out here. Okay, so it's a nice current orientation for MEG. The situation is a little different for the radial source, though. So here's if we now imagine the same field rotating around the radial source. It's not sticking out of the head nearly as nicely or really at all as compared to the tangential source. So if you have exactly this situation, then it is actually a current that is missed by the MEG sensors. However, we now know, so for a while we thought, oh, this is terrible, there's like this whole big blind spot, but it turns out there's actually very little activity that has this perfectly radial orientation. So usually the currents have a bit of a tangential component, and as soon as you have even a little bit of a tangential component, the field will start kind of sticking out of the head in a way that is better for our MEG measurement. So it turns out it's a very small percentage of the signal that has this perfectly radial orientation, and that MEG would be blind. So the blind spot is actually pretty tiny, but it does exist. Another consequence of the orientation sensitivity is that the signals are dominated by different currents in the two techniques, so EEG versus MEG. So that means that the two techniques kind of have different sweet spots. So EEG signal is dominated by activity in gyri, and so that's because it's perfectly happy to capture those radial currents, and those are the activities, or those are the currents that are actually most likely closest to the electrodes that are on the scalp. And in contrast, MEG activity is dominated by activity in sulci, so that's the visceral, the parts that go into the cortex or into the head. And that's because that's the cortex where you get that tangential orientation most easily for the current, and that's what MEG really likes to capture. So let's just take a peek at some cortex. So here, all these little arrows are representing neuronal current. And here we have our EEG electrodes on the scalp and MEG sensors outside the head. So they're sitting in their liquid helium. And so now, as you can see, in the visceral cortex, you get this really nice magnetic field that's easily captured by the MEG sensors, so it comes out of the head. And right here at the very top of the gyrus, you would miss these perfectly radial currents. But then EEG is not sensitive to the difference in the orientation. And given that these perfectly radial currents are here at the very top of the gyrus, and your EEG electrode might be sitting right on top of it, this would actually give a really, really nice EEG signal, whereas in MEG would be missed. So the signal can be dominated by different currents. So here is sort of the bottom line from these techniques. So EEG is cheap, it's widely available, and it offers excellent temporal resolution. It's a great technique for all those reasons. But MEG offers the best combination of both temporal and spatial resolution of all non-invasive cognitive neuroscience techniques. And that's a big deal because ultimately our models of cognitive functions in the brain do need to include both time and space. So if we don't understand the temporal aspect of processing, we don't really understand the process. And MEG gives us a way to get really good temporal resolution and then pretty good spatial resolution as well. And so for that reason, it's a powerful technique that does justify its price. There is one technique that's used with humans that rivals the resolution of MEG. And that is intracranial EEG. So this is not like the techniques that we've now talked about in this module because it is invasive. So this is a technique that's only available in a clinical setting with patients that are being prepared for brain surgery. So those patients will commonly have electrodes directly on their cortex that are being used to be able to plan the surgery better. And while those electrodes are on the patient's cortex, they also offer researchers the opportunity to run experiments with these patients, and therefore they can contribute to basic science in the course of the pre-surgical language planning, pre-surgical planning. And so that's a literature that has a better spatial and temporal resolution than non-invasive EEG, but it does occur in this very limited clinical setting. So now that we've surveyed the main techniques that we can use to study the brain, now we'll actually start learning about how we are using these techniques to reveal the neural basis of language, starting with speech.